Ribosomes are essential cellular structures responsible for protein synthesis, a process known as translation. They are found in all living cells, from simple bacteria to complex human cells. Ribosomes are complex macromolecular machines composed of two main subunits: a large ribosomal subunit and a small ribosomal subunit. These subunits typically exist separately in the cytoplasm and only come together when they initiate protein synthesis. When viewed under an electron microscope, ribosomes appear as flattened spherical particles with a diameter ranging from 15 to 25 nanometers. The two subunits fit together, creating a narrow cleft where the messenger RNA or mRNA molecule binds and is read. The large subunit contains three binding sites for transfer RNA or tRNA molecules A or amino acyl site, accommodates the incoming tRNA carrying a new amino acid P or peptidyl site, holds the tRNA with the growing polypeptide chain E or exit site, where the uncharged tRNA, having released its amino acid, departs from the ribosome. Ribosomes are ribonucleoprotein complexes, meaning they are made of both ribosomal RNA, or rRNA, and ribosomal proteins, or R proteins. Ribosomal RNA, or rRNA, forms the core of the ribosome structure and is crucial for its catalytic activity. The RNA molecules are highly folded and are responsible for forming peptide bonds between amino acids, a function known as peptidal transferase activity, making rRNA a ribozyme. These proteins surround the RNA and are believed to stabilize the ribosome structure and ensure the accuracy of protein synthesis. The exact number and types of ribosomal proteins vary between different. The primary and most crucial function of ribosomes is protein synthesis, also known as translation. This intricate process involves translating the genetic information encoded in messenger RNA or mRNA into a specific sequence of amino acids, which then fold into functional proteins. The process of protein synthesis on a ribosome can be broadly divided into three stages. Initiation. The small ribosomal subunit binds to the mRNA molecule and the first tRNA, which carries the start amino acid, usually methionine. The large subunit then joins to form a complete ribosome. Elongation. The ribosome moves along the mRNA, reading its sequence in triplets of nucleotides called codons. For each codon, a specific tRNA molecule carrying the corresponding amino acid enters the A site. The ribosome catalyzes the formation of a peptide bond between the amino acid on the incoming tRNA and the growing polypeptide chain held by the tRNA in the P site. The ribosome then translocates, moving the tRNAs and mRNA, so the uncharged tRNA moves to the E site and is released, and the growing polypeptide chain moves to the P site. Termination. The ribosome encounters a stop codon on the mRNA. There are no tRNAs that recognize stop codons. Instead, release factors bind to the ribosome, leading to the release of the completed polypeptide chain and the dissociation of the ribosomal subunits. In essence, ribosomes act as tiny protein factories within the cell, converting the genetic language of mRNA into the functional language of proteins. This process is fundamental to all life, as proteins carry out a vast array of cellular functions, from structural support and catalyzing reactions to transport. There are two main types of ribosomes, classified based on their size and sedimentation coefficient, which is measured in Svedberg units, also known as S. 70S ribosomes, also known as prokaryotic ribosomes, are found in prokaryotic cells, such as bacteria and archaea, as well as in the mitochondria and chloroplasts of eukaryotic cells. This resemblance to prokaryotic ribosomes supports the endosymbiotic theory. They are smaller in size with a sedimentation coefficient of 70S. These ribosomes are composed of two subunits. The large subunit, which is 50S, contains 23 sRNA, 5 sRNA, and approximately 34 proteins. The small subunit, which is 30S, contains 16 sRNA and approximately 21 proteins. In prokaryotes, 70S ribosomes are generally found freely scattered in the cytoplasm. 80S ribosomes, also known as eukaryotic ribosomes, are found in the cytoplasm of eukaryotic cells, including plants, animals, and fungi. They are larger in size with a sedimentation coefficient of 80s. These ribosomes are composed of two subunits. The large subunit, which is 60s, contains 28 srRNA, 5.8 srRNA, 5 srRNA, and approximately 40 proteins. The small subunit, which is 40s, contains 18 srRNA and around 30 proteins. Eukaryotic ribosomes can be found in two locations. First, free ribosomes are suspended in the cytosol. They synthesize proteins primarily for use within the cell, such as enzymes for glycolysis. Second, bound ribosomes are attached to the membranes of the endoplasmic reticulum, forming rough ER or the nuclear envelope. These ribosomes synthesize proteins destined for secretion outside the cell, insertion into membranes, or delivery to certain organelles like lysosomes, the Golgi apparatus, or the ER itself.